Oh, Amelie. Hey. So, thank you very much, all of you, for tuning in, and especially thank you to Emily for joining us today. Let me very briefly introduce Emily. So, Emily is Scottish. That's something we just learned while chit-chatting before this all started. And so, she did her undergrad in animal genomics at St. Andrew, and has been involved in all forms of quantitative animal genetics for all these years. She's quite well known in the field. And quite importantly for this channel, Emily has been very involved in projects like the Sheep Gene Expression Atlas project and these kind of things. And so these are important to us because they are typical of the kind of activities that take place in animal genomics, shortly following the same kind of activities in human genomics, And in many ways, the project through which I got to know Emily, which was Bovreg, can be somehow described as a kind of ENCODE for farm animal. And in the case of Bovreg, it was for cows. And so in this channel, we are very interested on one side in looking at what is going on at the forefront of human genomics. And last week we had Roderick Guigo, if you remember. I see you guys frozen. Do you hear me? Am I still live?
Yes.
Okay,
Yes,
good.
you are live.
So, sorry, yeah, because it's freezing from time to time. And so we had Roderick Guigo, who introduced the latest development in ENCODE and introduced these new 30,000 long non-coding RNAs. If you are following Twitter, this generated quite a bit of reaction from Salzburg and from a few people. And so I'm happy to say that the first announcement of the new 30,000 long non-coding RNA in ENCODE were on this channel. That's one of the things we're happy of. And so that's one thing, human genetics, the forefront. And the other one is the state of the art of animal genomics. And as it happens, Emily has taken the lead on a very important connection that is now taking place between FANG, this consortium dedicated to animal genomics for the benefits of genetics, and ELIXIR. ELIXIR, for those of you who are not familiar, but I'm sure Emily will introduce it, is a European infrastructure for data and to some extent for tools. And so now all of us are trying to build a community, a connection, a tighter connection with ELIXIR between FANG, especially EuroFANG and ELIXIR. And Emily is spearheading this effort. And so today, Emily kindly agreed to join us and to tell us a little bit about this ongoing effort and her vision about this new special interest group, I think is the name in ELIXIR. This is new community, sorry, this new ELIXIR community, and how this is going to be shaped. And you may want to mention your next step, Emily, or not. I don't know if this is public or not, or if this was just chit-chatting, but this is something quite relevant to all of us as well. So, Emily, thanks a lot. I'm taking also one parenthesis. Bjorn is leaving us. Bjorn is going to the pharma industry, so there's quite a lot of changes these days. And so Bjorn will kickstart this channel. I think it's the last time, maybe, or we don't know about your new attribution in your next step, but if you can stay with us, it would be great. But otherwise, I'm taking this opportunity to really thank you for your contribution all these years, all these months. Well, these years, actually, yes. And Emily, without further ado, the scene is yours. Thank you for being here.
Great. Thank you very much, Cedric. So I'm just going to share my screen. Hopefully this should be fine. Can you see that okay?
Yes.
Yes, great. Okay, so yeah, so it's lovely to see so many people on the call today, particularly others from the US as well. So today I'm going to not present any of my own research at all. This is really just an overview of the Domestic Animals Genome and Phenome Focus Group, which is a new focus group within the ELIXIR data infrastructure in Europe. So Just to briefly introduce ELIXIR. So ELIXIR is a European life sciences infrastructure. It brings together scientists from over 240 research institutes spread over 21 member countries and 3 observer countries, and also includes MBLEBI. So ELIXIR enables researchers to access and analyse life science data to improve the value and impact of life science research on public health, the environment and the economy. It provides guidelines and best practices for managing data, web portals, and has some approved data portals that we'll all be familiar with, for example, biosamples and also the European Nucleotide Archive. Also houses other compute resources, involves partnerships with industry and opportunities, crucially, to work together. And there's also small funding calls and things for ELIXIR members. So ELIXIR members and ELIXIR nodes are all within Europe. So ELIXIR was established in order to manage the huge amount of life sciences data that's now available. So there are several communities within ELIXIR, and these are groups of experts across the ELIXIR nodes. So the ELIXIR nodes are the different countries. Each country has a head of node. So the governance in ELIXIR is kind of well set out, for example, on the website, and probably not something we need to go into here, but I will mention nodes as we go through. So the communities represent scientific or technological themes. So you can see, for example, there are themes on food nutrition, metabolomics, microbiome, and maybe crucially for this community, biodiversity. So they drive the development of standards, services, and training across ELIXIR. And they provide an opportunity to be a part of a wide-reaching network of like-minded experts in this space, and also provide a mechanism for long-term collaboration with other ESFRI. So ESFRI is a term, a shortened abbreviation for the European Strategy Forum for Research Infrastructure. So there are several other large infrastructures in Europe, of which ELIXIR is one, and also other large-scale projects and initiatives as well. So these are the communities at the moment, and you can see that there's no community that's focused on animal science specifically, although it is encompassed to some extent by the biodiversity community. So there are also focus groups within ELIXIR. These are agile structures that bring interested parties together around a particular topic. So similar to communities, but not yet established, or not yet fully established, I should say. So typically they're used to address emerging areas of interest and seek to identify a strategy that ELIXIR can follow. So establishing a focus group can lead to a new ELIXIR community, and that's where we hope to go with the domestic animal genome and phenome focus group. But it doesn't have to be the default intention of a focus group. So focus groups are open to any member of ELIXIR, and plus where useful, external parties and other representatives. There are current focus groups at the moment, and you can see our focus group included here, which is domestic animals, genomes and phenomes. And there are other focus groups around pathogens, machine learning and fair data access, which are closely linked to ours. So one of the aims of the focus group is to work together with the other focus groups and make sure we don't overlap with our intentions and scope. So you can, through the talk, I've tried to put the web links in. So if you're interested in any more information on either communities, focus groups, or any of the other aspects, I've tried to put the links in the talk as well. And obviously this is being recorded, so you can go back and take a look at those in more detail. So our new focus group is the Domestic Animals Genome and Phenome Focus Group. It's a new focus group to support genotype and phenotype analysis for farmed and companion animal species. There's a link here to the focus group on the ELIXIR website. So it fits within the life sciences tier within ELIXIR. So ELIXIR has specific tiers based on its activities, and it fits within the strategic priority for biodiversity, food security and pathogens. Its ambition, the ambition of that scientific priority, is to mobilise and integrate molecular data to support transnational research programmes in biodiversity, food security and pathogens. So our focus group fits within that ambition, and the outcomes ideally for that strategic priority are coordinated and comprehensive activities built across ELIXIR communities that are relevant. So biodiversity, the biodiversity community, and also the pathogens in our community, the pathogens focus group and our focus group. So nodes are established as integral components managing national data, and nodes run as can run a suite of services allowing molecular cellular data and other types of data as long as it's shared in using fair data sharing standards. So there are collaborations and links to other disciplines and other infrastructures across Europe to facilitate seamless data management and connectivity. And that's the idea behind trying to merge across activities across communities and minimize potential overlaps as well. So our focus group addresses key ELIXIR societal challenges, including diminishing biodiversity, particularly in local and small populations of farmed animals, sustainable food production and the challenges associated with the growing human population, and also challenges associated with the changing societal perceptions around consumption of food products from animals. Also, the effects of mitigating climate change and the impacts of climate on farming and food production. And these all meet the key societal challenges around people, data, and science as well. So the goals of the focus group are to promote focus group activities, seek membership. So ideally, we'd have membership that encompasses across the whole of Europe, and scope future community needs in different ELIXIR nodes. So again, the ELIXIR nodes are different countries, really. So coordinate discussions and explore potential for data technology solutions for addressing key issues in welfare, behaviour, health, infectious diseases, metabolism and nutrition efficiency, as well as considering preservation of genetic diversity and the environment. The aim is to develop data standards, coordination workflows and visualizations for key developing areas, including pangenomics, functional genomics, genome editing, phenotyping and biobanking, which are needed to enhance data analysis and build on what we've already been able to do through, for example, EuroFang and the Global FANG Consortium, and also coordinating with other projects and efforts, and I'll mention those a bit later. So develop fair data management guidelines, standards and ontologies, and promote best practices in data coordination and archiving. And many of these things we've tackled already for the FANG Consortium. But ELIXIR has specific sets of these guidelines that can be adopted if we were to become a community. Also, crucially, organise tools and then data training and also knowledge transfer events. And there is some funding once we become a community available through ELIXIR to do that. So this is the leadership of the focus group at the moment. At the moment, I'm leading it because Peter established the focus group and then has subsequently moved on from his position at ELIXIR. So at the moment, the focus group's led by me and also Martin Groenen at ELIXIR in the Netherlands and Catherine Lazel at ELIXIR in France. And then we're helped a lot by Cecilia Chow, who helped to organise the meetings and share the materials for all of the meetings as well. So the focus group's connected to emerging genotype and phenotype research infrastructures in Europe, including EuroFang, which is a research infrastructure in its concept development phase to facilitate research and innovation for genotype to phenotype prediction in farmed animals. And then also to Phenome, which is a merging research infrastructure launched from a combination of previous species-focused research infrastructures, including AquaXL, PigWeb and SmartCow, developing phenotyping capabilities to support sustainable and resilient farming systems. And EuroFang and Phenome have actually merged shared efforts, I guess, are currently developing a proposal to become a project on the European Strategy Forum for Research Infrastructures Roadmap in 2026, and the merged infrastructure, which merges genotyping and phenotyping activities, will be called GenoPhoenix. So GenoPhoenix and Eurofang and also PhenoLive aim to improve efficiency, increase disease resistance, resistance to mitigation to climate change, and reducing environmental impact in order to meet the goals of the European Green Deal and also the Farm to Fork strategy laid out by the European Commission. And we aim to leverage the data generated through, for example, the Farm to Fork research strategy to meet these goals. And having a research infrastructure that addresses these things is hugely important and identified as a gap. So a research infrastructure that provides a consolidated infrastructure for animal science in Europe that fill the gap identified in the last European Strategy Forum for Research Infrastructures roadmap. Okay. So the Eurofang Research Infrastructure Project, which was really the basis for the focus group, is funded through the InfraDev funding stream through Horizon Europe for developing, consolidating and optimizing the European research infrastructures landscape and maintaining global leadership in this space. It has 7 different partners and it runs until the end of 2025. So we just got about a year left on that now. And it's coordinated by FBN in Germany and co-coordinated by the University of Edinburgh. So as part of Eurofang efforts, we developed the focus group for domestic animal genomes and phenomes, and it's really designed to help facilitate the creation of a common data access and data structure across Europe, supporting and providing hopefully additional support for the FANG data portal, and also to expand the data portal around curation and biobanking of in vitro cellular systems, and also integrating phenotype information from, for example, the PhenoLive research infrastructure, the new research infrastructure GenoPhoenix, and other efforts for farmed animal phenotyping in Europe, including the EU Life Cost Action and existing infrastructures for phenotyping. So we aim in Eurofang to connect with existing projects and infrastructures and consolidate G2P research in this space, both in Europe and also globally. So with global initiatives such as AgBioData and AG2PI. So global collaboration is really central to the focus group and also Eurofang more broadly. So this is a hypothetical example of what the Eurofang research infrastructure aims to do. And this example is kind of an idea of how the focus group needs to be able to show what the community needs in terms of access to data, access to specific components of achieving a research goal, I guess, that are related to data curation and data access. And that can take many, I guess, forms. So this is an example identifying resistance alleles for a viral pathogen using high-throughput CRISPR screens. So a researcher has an idea, they work on a target host species, and they have access to a target pathogen, but they don't have access to in vitro cellular systems that are suitable. So they make an access request to the infrastructure partners, including, for example, INRA or Wageningen. So they ask whether they can utilize those in vitro cellular systems. They also want to utilize libraries of sgRNAs for genome editing to do high throughput CRISPR screens. And they would ask Roslin whether they could access those through the infrastructure. We know from concept development for the infrastructure that we need to build more capacity to do high content CRISPR screens more broadly in farmed animals. So that's one of the things that we've been developing through, or the capacity to do this, trying to develop that through the infrastructure. They can make sequencing requests through other infrastructures such as ELIXIR or commercial providers, but crucially for the focus group, they would also need to curate the data, and that would be a community need that they could access the data through the data portal. They can access all of the data, for example, from the Horizon 2020 projects, such as the Gene Switch project for pigs, so all of the regulatory and gene expression information. But they need to do that through a curated way. It's not very easy to just find that information in the ENA or in biosamples, for example. So these resources are managed through EBI and through ELIXIR data resources such as the ENA and biosamples. So So the concept for the Eurofang research infrastructure and how it could be accessed by researchers is really underpinned by access to data. And these are the things that we want to explore through the focus group, because there are many different research questions, obviously, that the community is asking of the data. So the idea behind these new infrastructures is to meet the current challenges and demands in the agri-food sector, integrating European facilities for phenotyping, biobanking, imaging, omics data, and veterinary facilities at the farm and environmental farm scale and laboratory scale, I guess, in terms of fundamental science, integrating approaches, including e-infrastructures, to systematically predict, diagnose, and prevent animal disease, which is key. So ELIXIR resources can be leveraged to do that, but we need to say what the community needs to do it. So PhenoLive is based around phenotyping, whereas Eurofang is really linking genotype to phenotype. So this is the concept for PhenoLive. It's based around generating high-resolution phenotypes for complex traits in farmed animals. And the ELIXIR components, sorry, the ELIXIR components are based around large data sets and sample databases, and these should be shared through fair data sharing standards, but that's a bit more difficult for phenotyping data. And one of the things we're exploring with the PhenoLive partners in developing GenoPhoenix is how we could expand, for example, the data portal for FANG to leverage these more commercially sensitive data sets, and develop a European distributed multi-species and multidisciplinary RI that manages this kind of data, as well as the data, for example, that's available through the data portal at the moment. And there are links with current European cost actions such as EU Life to be able to do that as well. So the aim is to merge rich and existing standards and scientific knowledge. So the Global Functional Annotation of Animal Genomes Consortium, or FANG, has been doing this for the last 10 years or so, developing rich, fair standards for animal genotype to phenotype community. The FANG and Eurofang data coordination centres based at EMBL already have strong connections and interoperability with ELIXIR resources, such as the ENA and biosamples that can now be further strengthened through the focus group. And it would be even further strengthened if we became an ELIXIR community. So FANG and Eurofang are engaged in active communities, and we work really well together, I think. And they desire to make data easily accessible and collectively improve coordination and analysis methods towards a shared goal. We have lots to share with and learn from other communities, including other ELIXIR communities, particularly bringing genotype and phenotype communities together with a focus on best practices for data management, data exchange and analysis. It's very important through the focus group, and what the focus group can do is to consolidate community resources and objectives. So, for example, there was a really nice seminar by Guillaume de Valle recently on the BizFada platform for visualization of FANG data. AgBio is very active globally, and particularly in the US, around enhancing research outcomes for data across agricultural species. And then a colleague, Emmett Tannen, mentioned to me just yesterday the vertebrate breed ontology, which has just been, I guess, updated and released as a preprint as well. So it's trying to consolidate all these efforts together and have a place where they can be kind of consolidated and work towards a shared outcome. But there are many other ongoing projects and communities, including the various pan-genome consortia for farmed animals, the telomere-to-telomere genome consortia, and this should hopefully help to bring these global resources together. But for ELIXIR, there is a focus to some extent on the resources available in Europe, but they need to be accessible also to the global community. So we're building on our strong connection to the NF core communities, and one of the reasons I think Cedric and Bjorn and Jose invited me to talk today is the link with the NF core community for animal genomics. So over the last 4 years, there were 6 Eurofang Animal Genomics Horizon 2020 projects that have collaborated on development of genomics pipelines in NF core, and they, I guess, developed into the Animal Genomics Special Interest Group for NF Core, which brings together developers working towards collaborative genomics analysis tools. So for the focus group, our aim is to collaborate with this group on standards, best practice, and wider adoption in the wider domestic animal genome and phenome communities. These are the, obviously probably everybody on this call knows the details of the special interest group. So what do we need to do to become a community? That's the key thing for the focus group. And becoming a community is a very useful thing politically in Europe. It shows the European Commission when we put in funding applications that there is a networked and really active community in animal science broadly, I think. So we used to develop a white paper and a roadmap, and this is an example on the right of the ELIXIR Biodiversity Communities white paper. So all of the white papers and roadmaps are published in F1000 research so that they can be continually updated by the community. So currently we have a white paper in a draft form, which we plan to present to the ELIXIR heads of nodes for consideration as a community in 2025. So what we need to do is produce this white paper and roadmap. They don't have to be final, but they do have to be quite a long way to becoming a very clear vision of what the community would look like. So the white paper needs to set out the challenges faced by domestic animals at the moment, so the global challenges and priorities, and then also the scientific and bioinformatic priorities to do domestic animal science, and then also outline the opportunities that could be provided by ELIXIR. So if you're interested in what this might look like, the biodiversity community white paper is really excellent and really gives a good overview of the content of what would be necessary for the white paper. So at the moment for the white paper that we're developing, we're specifically focusing, for example, on the challenges to farmed animal food production, the resources that we currently have, the FANG data portal, the work done through the Eurofang and PhenoLive research infrastructures and Genophemics, and trying to look across Europe at the other infrastructures and how that we can work together with those. So this is a sort of ongoing effort. We have monthly meetings for the focus group every Monday, first Monday of every month at 2 p.m. In Europe. And to join the mailing list for the focus group, you can go to this address or also contact Facilia as well. So the focus group is open to really anybody that would like to join. I would like to probably, it sort of would be great to get as much across-country representation in the focus group as possible. So for example, we just had a member join from the Czech Republic, which is great, who was focused on functional annotation in zebrafish genomes. So there's huge scope to try and get as much representation across ELIXIR nodes as possible. So we can contact Facilia as well to be added to the meeting invitations. So just to say the work we've done on the focus group so far has largely been funded through the Eurofang Research Infrastructure Grant. And these are my contact details and also more details of the focus group online. So thank you everyone for listening. I'm happy to answer questions about it or provide directions for sourcing other information. Thanks.
Thank you very much, Emily. This was very informative and I believe very interesting to this entire community. Just while we wait for questions to turn up, just going back on the introduction I made. So this special interest group is dedicated to animal genomics scientists using NF core. Oh, my camera is, let me just try something. Switch my camera on and off. again, and let me try again. Oh yeah, I'm back. So my this special interest group is dedicated to scientists, to animal genomic scientists using NF-core tools, and so we have an ongoing reflection on how to use this tool, what are the current gaps, how does the community, what kind of tools will the community need, and Emily was kind enough to invite us to attend the preparation of the focus paper for our contribution along these tools. And so one of the things we have discussed with Emily and with the focus group is to leverage this NF-core community so as to bring to the paper the focus of the NF-core community. Of course, NF-core is not the only available collection of tools, but I believe it is one of the most formally constituted community for genomics analysis, especially at the high throughput level that is required for animal genomics. You know, there are more user-friendly tools and this kind of things, but when it comes to do high throughput command line analysis, this is probably the most usable resource. And so in the months to come, we will have an ongoing reflection on this channel so as to best contribute our vision to, or propose our vision to Emily for the focus paper. Now, does anybody have any question? I have quite a few actually, but if any of you want to start,
Brenda has a question. Raise her hand.
Yes.
Thank you, Emily, and thank you for the invitation. My main question is really how can folks in the US more specifically join and participate in this? As you're aware, you know, we have you mentioned a number of the ruminant T2T pangenomes and FANG initiatives that are here in the US. So if you can just address that a little bit and let us know how we can become more involved, that'd be greatly appreciated.
Sure, thanks, Brenda. So yeah, so one of the key, I guess, components of this is making sure that the community captures efforts that are undertaken globally. So as you mentioned, many of them, and obviously developing the tools and managing the data that's coming through through ELIXIR resources such as the ENA and biosamples and things is critical. So I think in terms of the white paper, it would be very useful to have input from colleagues in the US to develop a specific, I don't know if it's the right thing to do to develop a specific section on global, on efforts outside Europe, or to try and just integrate those across the focus group, across the white paper, because they're all working towards a consolidated and shared vision for animal science. We do have to have a focus in the white paper on European, I guess, activities, but that's more of a political kind of a thing. And I think that integrating the global activities is crucial specifically in our space. So there's opportunity definitely to join the focus group, and it would be great to have representation of the main projects in the US in the focus group, I think. But we'd try and present it as an integrated approach, I think, rather than these are the US projects and these are the European ones.
I hear what you're telling me. Thank you.
Great.
And can I just add, at a much lower level, the NF-core Special Interest Group is also here to provide the community of scientists all across the board a channel to discuss harmonizing procedures. You know, this is typically the kind of place where people can discuss how do you call your SNPs, you know, how do you do your ATAC-seq, and this is meant to have a kind of bottom-up emergence, community-driven standards, which I believe will contribute to this. So, like Emily was saying, you get Eurofang, there is a little bit of European politics there, but overall, if we work nicely together, we can have a very solid, consensual standard that will emerge, thus making our data entirely interoperable. I think, you know, at our level, at the NF core level, this is really our aim, making sure that our data is comparable across as many places as possible. And so if you are interested in contributing to NF core, this special interest group, so it's this, I see a lot of new faces, and just to make it clear, on one side we have the focus group, which is this Eurofank focus group, and that's very general. This covers all aspects of genomics. And at a much lower level, we have this NF core special interest group, which is part of NF core. And that's where we discuss specifically Nextflow based tools. That's just this articulation. And so we are, trying to contribute to the tool part of what Emily is doing. I think, Christopher, you were raising your hands. Probably you have a question.
Yeah,
If you can
thank
unmute
you.
yourself. Yeah.
Thanks. So I'm interested in advancing single cell analysis. And so I was wondering if in your special interest group in NF core, are there people working on pipelines for single cell analysis, cell annotations, that kind of thing? Or is that something we should build?
Go ahead.
Just going to say,
Anybody
Cedric,
wants
it's
to?
more related to the bioinformatics pipeline. So Chris, just to answer at a high level for the focus group, it's more just addressed at a higher level in saying that we've got these resources for single cell genomics that need to be maintained and developed, and through ELIXIR, for example, that would be Array Express that's maintained by EMBL-EBI. And there's a new team leader at EMBL-EBI on functional genomics who's developing the Array Express protocols, and probably would pick up on where you left off with Peter, potentially on that, if you had, I can't remember the grant that you had. So there's ways through ELIXIR services to manage single cell, but it's at relatively, in terms of the white paper and the focus group, it's at quite a high level. But Jose or Cedric maybe could talk about the granularity at the pipeline level.
Yes, we have. This is typically by its origin, Nextflow and NF core are very focused on genomics activities, and so we have an NF core pipeline, which is a single cell RNA-seq pipeline. I've just pasted a link. We also have under very active development a differential abundance pipeline that we hope to better connect with a single cell RNA-seq pipeline. And one thing that is quite important to comprehend is that these pipelines are community developed pipelines, and this is explained actually in this recent update of NF core that is under review in Genome Biology. It goes out at some point, but there's a preprint somewhere. Jose, maybe you want to cut and paste it somewhere. And so these pipelines are pipelines we collectively maintain across sites, and their updates are validated by the community. Their testing is carried out automatically by the community, so that industry standards are maintained all across, which is quite interesting and important and unusual for academic pipelines.
Okay.
So does this answer your question, Christopher?
And
so if
yes.
you wanted to actually, yeah.
Yes, I guess there is a group in the U.S. covering plants and animals that is interested in that. It's part of Ag Biodata, and Peter and I started that. So there would be interest in seeing, not only the initial data analysis, which I guess I don't know what exactly is involved in your pipeline, but one of the big issues we see is there's many techniques and different approaches to collecting the data, and they all have different analysis tools, which they don't all talk to each other very well. So it's just a starting point that we're discussing. How do we try to work together, and how do we try to create something that people can use?
Exactly, you're spot on. And so it's full of methods. It's full of different ways of slicing and dicing the data. They all have their pros and cons. We know this, and this is why one of our focus in NF core now is to build what we call method hubs, hubs in which all the alternative ways of analyzing similar data are put together and made as interoperable as possible, so that you can objectively say, you know, here is my input. That's the kind of things I would expect. First of all, what is going to give me the best value for money? Because, you know, maybe you want to be as accurate as possible. Maybe you also want to deal with a very large amount of data for a reasonable price. So all of these questions should be asked. And a very important aspect for you is that if you're interested in that stuff, you can just contribute. All of these pipelines are open. You just have to become a member of the community and you say, okay, we want that method to be put in. You go to GitHub, your guys figure out how to plug in the tool or the combination of tools they are interested in, and that becomes part of the pipeline. And that's a very important aspect, that all of these things are open and are meant to grow, but they are not meant to grow without any control. And that's why, you know, any pull request has to be approved by the core members. Jose Espinosa here and Julia Mier, who is on the call, are part of the NF core team member. And so their contribution is to keep an eye on the pull requests made to the pipeline and making sure that the pipelines are growing the right way. Okay.
I've seen the comments in the chat as well. Thank you.
Okay. So that was me pitching NF core. Sorry, I got excited. Any other question for Emily? I have one. Then while we wait so interrupt me if somebody has one. But I have one. So you know, one of the things that came across when we were working with the BOVREG data is that I realized that animal, you know, as much as human data is protected for reasons of privacy, animal data is protected for reasons of economic value. And that often raises issues because I still remember a long time ago in Phenome, which was another project, we had this extraordinary table. And suddenly we realized that the interesting column, which was food efficiency, the column on which we could have made some interesting correlation was not given to us because this was a very valuable column. And it seems to me that this is a recurring feature in animal genomics, that the data is generated with a very specific commercial purpose in mind, and its access can be restricted. How do you think this can be dealt with, or do you see any way of having because we know that in human health, you know, a consolidated data set unlock everything. And so how do you see this happening on the Eurofang side?
Yeah, so we have tried to explore this with Eurofang through one of the work packages in the infrastructure, and have so we invited Hao Cheng at Iowa State to talk about data encryption and how you can encrypt commercially sensitive data prior to sharing, and that seems like one really obvious way to do it, which has been developed, I think, through funding in the US and through the AG2PI program, sorry. So that his ideas seem really exciting, and the ideas of others in that space to be able to then potentially share commercial data without compromising IP. So we talked a bit with the other phenotyping infrastructures. I see Claudia's on the call as well, and particularly the PigWeb infrastructure, about how phenotyping data in particular is shared at the moment or made publicly available. And it really doesn't have a kind of consolidated approach to sharing phenotype data. Much of it ends up on Zenodo, for example. And I think we need to explore how the FANG data portal could be expanded. And one of the things with the new merged GenoPhoenix research infrastructure is how to do that with the phenotyping community and all of the potential issues that might involve. We've had many discussions over the years, I think, about partitioning part of the portal. In BOVREG, we had the part of the portal that had industry data in that only BOVREG partners that were working on it could see, but it could be shared across partners at least. So there are ways to do it, but it is difficult, I think. and obviously will be one of the components in drafting the white paper as to how we manage that, because it's crucial in linking genotype to phenotype, I think.
Thank you, Emily. I mean, I really have a vested interest in this because if you remember, this group started as a farm animal Eurofend tools working group, and when we moved it to NF Core, we had a long heated discussion, and we dropped the farm, and it became an animal genomics group. And the reason behind this is very, very clear. It's because of the Earth Biogenome project. This project is going to massively power all kind of genomics, with the diversity. We know this because if you look at the pan-primate genome, just the sequencing of all of our cousins is bringing much more insight into medicine than any of the very sophisticated SNP calling, SNP pathological SNP prediction that were made before. Just straightforward comparison. And so all of this data is about to arrive. We were supposed to have Harris Lewin, the chair of the EBP, talking last month, but unfortunately Harris was sick, so this has been rescheduled for next year. And we are very excited to, in the NF Core, asking ourselves, how are we going to make all of this data interoperable? How are we going to make sure that all of the species can shade light onto one another? And from that perspective, the tool, common procedures, common ways of using the tools, procedures that are reasonably robust across enough species is something, it's a very fundamental aspect we have to explore as well. So yes, this interoperability it goes between species, between analyses, it goes all the way. And Harris likes to say that ENCODE was just a pilot for FANG and for everything that, it was a small scale pilot for everything that is now going on in animal genomics, and in many ways it was. Do we have any other question for Emily? I have another one. So it's quite refreshing these days. Oh, I see some messages maybe on the Slack. Maybe there's a question. Some comment by Chris about the NIFA project funded to look at data federation and encryption. Yeah. And I know that this is also a very big deal in human genomics, right? Because you want, unfortunately, you cannot anonymize genomics data. You are your genes. And so the minute your genes are put online, you are put online. But the ways of encrypting the data so that interesting properties can still be analyzed without anonymity being compromised are an active field of research. Yes, I was going to say, it's a long time I had not heard the word AI in any talk, and so you did not mention AI, but surely some AI will have to be implemented here and there to leverage the data to help setting up protocols and standards. Have you started thinking about it a little bit?
Yeah, I mean, yeah, that was maybe an oversight to have not mentioned AI, but ELIXIR itself, I guess, was partially set up to accommodate the use of AI, or to preempt the potential applications. And I can't remember whether there's a specific community focused on AI, or whether it's addressed across the communities. But for example, so I was in the cellular and molecular resources community, where it's a key component of their agenda, and I think it does have to be a key component of the focus group, and the opportunities that it provides are just huge, and in also in terms of managing the data, I think, and being able to utilize it. So another reason to make the metadata as good as possible, so that it then actually is useful for the AI tools. So I think often the biggest challenge with AI is actually using AI using the data for that purpose, because it just hasn't got the appropriate information. Yeah. So yeah.
I can tell you what was my most frustrating, the most frustrating part for me on BovReg and all these things which is it's the ontology. I mean, you
Yeah.
know that the ontologies are the solution to everything. You know that if things are named the right way, you can programmatically do about anything. But naming things the right way is extremely resource intensive. And at the end of the day, we all prepare the layout and say, if we had the ontology, this and this and this would work, but then somebody has to do it. And I have the impression, and you'll correct me if I'm wrong, that well-managed AI could unlock the ontology issue, that it could, relabel everything that is trivial to relabel and generate ontologies based on the metadata.
Yeah, I think so. I mean, so we have an advantage in that we've developed these very specific metadata standards already for FANG, which is helpful. But yeah, I agree. So for example, for the Farm GTEx project, where they pulled in so much expression data across so many projects, having an AI tool that would help to, like you say, correct the wrong entries or the non-entries, I suppose. And I think actually maybe the Farm GTEx projects are even looking at how you can pull, for example, the sex of the animals from the gene expression data if it's not included in the meta-analysis, and whether that could then be linked to AI tools as well. So there's huge potential to develop that further and add missing information.
So maybe then going back to our American colleagues, I get to know some of you almost 20 years ago during the Quantumix project that we were running, because at the time the EU was putting, the EU and the US have a joint interest in food security, and the EU was putting quite a lot of resources in making sure that bridges will be built between the two continents, between these two major markets. And what is the situation now? So are you guys putting together the US equivalent of Eurofang, or is there is it something, Chris asked Emily the question, if we all were collaborating together, is it something that could get support on your side, or is there any way going this way? Maybe Brenda or Chris?
Well, there's a number of things going on. Probably the focus right now is there's a funded project in agricultural genomes to phenomes, and we developed sort of the community there globally, but again, it was more focused on U.S. interests in agriculture, but plants and animals. There's not the same type of sort of community building infrastructure, I would say, in the US as there is in European community.
Okay. So this year we are looking around to see if NF-Core members will be at PAG, but we will not have a formal representation of NF-Core at PAG this year. But next year, for 2026, we are planning to put together a workshop, probably an NF-Core workshop, where we'll get a chance to discuss with our American colleagues and anybody going at PAG, some potential interaction around NF-Core. So if any of you will be interested being part of this effort, please reach out to Jose or to thanks Brenda. So reach out to Jose and to me, and we'll put you in the loop.
I would just do a quick advertising for at the AGBT Ag meeting in the end of March. We're going to have a single cell workshop, which is going to be focused on community building and data resources discussion. So that might be something people are interested in. It's been advertised briefly. We don't quite have registration set up for it in terms of the program and so forth, but that's hopefully coming within the next week.
That's interesting. Please send us a pointer, Chris, and we will diffuse it on the NF-Core list.
The other thing that I can share is we did get funding for an animal genomes sort of network for farm animals. And that in that funding, it has sequencing initiatives, it has FANG initiatives, it has precision agriculture initiatives, it has 3D genomics initiatives. And the idea behind that network funding is to bring people together to forward some of these initiatives. So I would say that it seems to me that the FANG in the US, what was put forth has kind of, I guess, to kind of is not continuing to be funded or to go up. So, and the reason why I mentioned this animal genomics sort of network is if folks in the US want to get together in those spaces and to raise attention to our funders or our funding agencies, NIFA and NSF and those other types of things, this might be a good avenue to do that. I think as Chris has mentioned, we don't really have any of those priorities set, but as the new administration comes through, new priorities will be coming forth. And one of our USDA NIFA representatives, we have to rewrite our 10-year plan for the USDA. And I think this is also a good place to plant these seeds and to talk about how we've put all these resources in this and how to move that forward instead of, okay, we've done it, check the box and move on to some other mandate or some other priority that gets dropped in. So if we want to move together and ask for our funders to consider putting out calls, that those would be the opportunities to do that. I don't know what
Brenda, if I can bounce on this. So a real issue of all these things, and this is true of Europe as well, is continuity of funding. You get funded, you do stuff, you stop being funded, the infrastructure dies, and you have to revive it. And so ELIXIR was an attempt to address this problem by providing a more stable framework, a long-term framework, because this was recognized. I'm old enough to remember that Swissprot was in crisis every second year because suddenly the funding was gone and that was it. And then it was. So one of the things I'd like to stress, and to me that was very important in BOVREG, is the sustainability and the continuity. It's not like you sequence one thing and that's done deal and you check your box. And no, this is an ongoing effort. And the pipelines are, like the name indicate, they are tubes through which data will keep flowing. From that perspective, NF-Core is a very sustainable resource in the sense that if you contribute to NF-Core, if your funding dries out, what you have done stays there, it stays alive in the community, it keeps being maintained by the community. And when your funding comes back through other channels, you can pick up your work where you left it, or even where it has been taken a little bit further. And that is something I think very important to point out to founders, is that we have a live infrastructure. It's 15,000 people now working on NF-Core and collectively maintaining this infrastructure. So it's a lot of people. They're very dedicated. There is also industrial support. There is this company called Secura Lab, which is actually helping maintaining the infrastructure. They sponsor a bit of the infrastructure and a bit of the conference. But the bulk of the infrastructure is really provided pro bono by the community, by all of us, helping this and that pipeline and this type of things. And we are now going to start raising a little bit of money for NF-Core, applying for grants and this kind of things. But the long-term vision is a resource that is sustainable through the continuous community commitment. And so if you guys want to piggy ride on this, that's exactly what it is meant for. It is meant for anyone who wants to make a contribution, to hook their contribution so that it becomes live and plugged in a broader framework meant to remain alive. It's already 1 hour, so, and I'm told by my neuro friends that this is as much as the human brain can sustain Zoom, otherwise neurons start melting. Jose?
Yes, maybe before wrapping up, just to announce that next meeting on the 15th of January will be devoted to actually work in this section of the white paper about the technical part. And all of you are very well welcome if you want to be around and help discussing this part of the paper, of the white paper.
Yes, and we will try to engage some of our NF Core colleagues to join as well, even if they are not in farm genomics, because my main line of work is evolutionary biology. So I don't see any real difference between human and animals. As long as you can align it, it's good enough for me. And so we'd like to have this as broad as possible. And so before we wrap up, thanks again to Bjorn for being a big part of kickstarting this group. And I hope we see you again on this channel in the near future. I know we will see you on other NF Core channels anyway, but in your next step, I hope we also see you again. And if there is nothing else to say, then I wish you all a great holiday season, and I hope you have a great time, your families and friends over these next weeks, and that you come back full of energy and refreshed and ready to work on. Oh, Jose, this is impressive. Okay, and so thank you all for being. We had up to 35 people today. Is this correct, Jose?
40 at the
40 people. Emily, we'll have you sent a record. I have had the highest
Oh, maybe
on the special interest group.
yeah, maybe it's just everybody's got a free time before Christmas. You never know.
Yeah,
Oh,
nobody has anything else to do the week before
exactly.
Christmas. No,
Possibly.
that's not true. Everybody's
Oh,
absolutely busy these weeks. So they
yeah.
came because you had something interesting to say. Thanks a lot, Emily. And
Thank
to
you.
you all, thank you very much and see you very soon. Bye-bye.
Bye bye.